Hey, it's Professor Gould again. In this section, we're going to learn how the cell regulates going into the cell, these parts of the cell cycle, and moving from one to the next. Because you can't just be doing this willy-nilly. All right. There are two categories of regulatory proteins. This is going to really surprise you. One set, positive regulation proteins. They promote the progression to the next phase. Guess what the other one's called? Negative regulation. I know, right? Okay. So they block progression to the next phase. So these are going to be active during those checkpoints. Now, positive regulation is going to depend primarily on, remember all that cell communication stuff we talked about last week? Yeah. Okay. Here we go. A protein called a cyclin, which is shown here in this green blob, binds to a cyclindependent kinase, which is the blue blob, activates the cyclindependent kinase once this kinase binds to the cyclin. Because the cyclin, the kinase, is dependent on being bound to the cyclin in order to be activated. That's why it's called a cyclin-dependent kinase. Okay. You remember that a kinase protein is one that moves a phosphate from one place to another. That's what the kinase in the name means. This is not my favorite. I feel like they should be phosphatases, but they're not. They're kinases. And so in this diagram, this pink blob up here, that's the ATP. Okay. So the kinase, once it's activated after binding to cyclin, it can then hand off this ATP to another molecule to phosphorylate another molecule. Together we call this a cyclin CDK complex. All right, so this is what it looks like. Here's this cyclin-dependent kinase. Here's the cyclin hanging out, waiting for a kinase to come bind to it. They bind together, and then the kinase binds and transfers a phosphorus. Now, the cyclin-dependent kinase can transfer that phosphorus to another target protein. So this kinase is going to have two binding sites, a kinase site and a target protein site. The phosphate group gets transferred, then unbinds from the cyclin-dependent kinase, and boom, is now phosphorylated and can go on to do another part of the cell signaling pathway. Okay, so this is part of a signaling pathway. This is then going to be this is now an active protein, and then it's going to go do stuff. Okay, we're not going to get too much more into the nitty-gritty of it. What we will say is that there are actually 4 different types of cyclins named A, B, D, and E. Don't ask me why there isn't a C, but they are each more prevalent during different phases of the cycle. So these are going to be made at different points in the cycle by the cell. So it's both, this is responding to internal and external signals in the cell. Okay, so this cell could be responding to a signal from outside, something binding to a signal protein on the membrane, or responding to some pathway inside the cell. Okay, both of those can make these happen. And then as long as these are being made in the correct time, then everything proceeds fine. But imagine if you had cyclin B, which is supposed to be really high here as the cell goes into mitosis. Imagine that it was high at the end of S phase back here. The cell would skip G phase. It needs G phase before it can do mitosis. But if cyclin B is made at the wrong time, then the cell's just going to go do mitosis without making sure it's big enough. And then you're going to have all kinds of problems. All right, so what about the negative regulation protein? So this cyclin-dependent kinase, this is the positive regulation, okay? Promotes movement into the next phase. Negative regulation is going to say, whoa, hang on. Okay, so remember we talked about the checkpoints. So these are part of that checkpoint process. There's a bunch of proteins. Okay, there's a whole group called retinoblastoma proteins, or RB. They were discovered in a cancer in the retina in the eye. So that's how they got their name. They are not only present in the retina, they're actually common throughout the body. There's also P53 and P21, which are named for their molecular weight. So there you go. Mostly these are active during the G1 checkpoint. Remember, the G1 checkpoint is our primary regulatory checkpoint, determining whether a cell is going to go into S phase and complete mitosis or not. So RB is going to monitor the size of the cell. Is it big enough? Once it's big enough, RB will allow it to go to the next phase. P53 is going to check the DNA, recognize damaged DNA, and either trigger DNA repair or apoptosis of the cell. If the DNA is really, really messed up, the P53 can just start apoptosis. And then P21 is caused by a rise in P53, and that if P53 levels are high, P21 levels are going to rise, and that is going to stop the cell in that G1 phase. Okay? These are all called tumor suppressor proteins because they are responsible for stopping mitosis. A tumor is just the result of a cell or cells doing mitosis too much. It's a result of a failure of the cell regulation machinery. So instead of stopping, the cell's just going to be like, oh, mitosis. I'm just going to keep doing the cell cycle uncontrolled and just keep growing. Then you have a mass of extra cells. That's called a tumor. Might be dangerous, might not. A mole is actually a tumor. Just a tumor that stopped growing at some point. I know. Yeah. If your moles don't stop growing, get them checked out. All right. What about cells that are doing mitosis all the time and are supposed to? We talked about the basal cells in the integument, those cells at the bottom of the epidermis that are supposed to do mitosis so that they can replace the surface cells that die and get sloughed off. There are also mesenchymal cells that make all of your connective tissues, your tendons, your ligaments, your fat cells. The cells in those tissues, tendons, ligament, fat, they don't themselves do mitosis. If you need new fat cells, and yeah, you can totally make new fat cells, you don't do mitosis of a fat cell. You do mitosis of a mesenchymal cell that makes a new fat cell. Okay. There's also cells lining the digestive system. Your digestive system is a really harsh environment. So the surface cells, again, die and get sloughed off. So there's special cells that do mitosis and replace those. So why would those cells keep doing mitosis when others can't? And if you're thinking, well, they must have less of those negative regulatory proteins. Yeah, that's exactly it. They have enough to do the checkpoint, but not enough of, for example, P21 to stay at that checkpoint. That's kind of cool, isn't it? Right? So they have just enough to make sure that the cell does the whole life cycle before doing mitosis, but not so much that the cell gets stuck in G1. All right. What about cells that never do mitosis? You're thinking, are there cells that never do mitosis? Well, I just told you about the fat cells, didn't I? But also your neurons. Your whole life, you have the neurons you had when you were born. Yeah, you tend to not make any more, except in the actual brain, which does grow during childhood. Once you reach adulthood, no. Except maybe in a couple special areas. Okay, it's complicated. There's a couple special areas in the brain, you might make some new neurons. Spinal cord, you are not making new neurons in the spinal cord. You kill one, it's dead. That's just the way it is. You might be able to repair it, but you're not making new ones. What else? Most of your skin cells, once they're made, they don't do mitosis. The basal cells do mitosis, but once they make a copy, that one doesn't do mitosis. So if there's a cell that's not going to do mitosis, where would we expect it to stop? We would expect it to stop here in G1. And what it does is it actually exits interface and goes into a thing that we call G0. And G0 is like, okay, I'm so out of the cell life cycle, like I'm never doing mitosis, so I don't need to even think about all of this. It's like a permanent G1 regulatory stop. So the muscle cells in your heart, yeah, they don't do mitosis. You need new heart cells. You're going to have other cells to do mitosis and make new ones. Your neurons, your fat cells. And also, for some cells, they're going to like have a momentary or a temporary move to G0, but then they can get out of G0, go back to G1, do the life cycle. So thinking about the cells in your liver, most of the time, those cells don't need to replicate. They're doing their thing. Yeah. They're hanging out here in interphase, right? Doing their little liver thing, making bile, breaking down toxins. But then if there's damage to the liver, your liver can actually regenerate. And so some of those cells are actually going to pop back into G1 and complete the cell life cycle. Once everything has been repaired, they will pop back out to G0 and go back to being, their liver cells. This is how T cells, those memory cells that we talked about in the immune system, that's what happens to them most of their lives. They're often G0. Once they get activated, they pop back into G1, go through the cycle to make copies of themselves, to make a clone, to produce antibodies, which we call clonal antibodies. Yeah, they're antibodies made by T cells. I know, memory T cells. So if you want to just like have a bunch of clonal antibodies at the ready, get vaccinated so you can make some T cells so they can become memory cells so they can make antibodies when you need them. That is so cool, right? All right. We're going to pause there. And next up, we're actually going to do mitosis.